Okay, here's the book I've been reading. It's called The Reference Manual about Rosenow et al. So this guy, Carl Rosenow, he was a real famous doctor back in his day, the early 1900s, and he was the head of bacteriology, experimental bacteriology at the Mayo Clinic. And he was considered a genius, and a lot of people were very impressed by his work. He was a pioneer of the idea of a focal infection theory, that you get an infection in your tooth or your tooth root, the jawbone, so to speak, and it can then spread infections to other location. And it correlated very well with the work of Weston Price, focal infection theory, whereby they would take the bacteria out of the human's mouth who got a subsequent endocarditis of the heart, kidney infection, or some other form of infection, brain abscess or something, implant it in underneath the skin of a rabbit, and it would get the same infection. Okay, so anyways, he's one of the pioneer guys in this, and he developed all sorts of techniques to culture bacteria in different ways, to dilute them, and to confirm where the origin was, coming from oral bacteria. Okay, so I'm going to show you there's some pretty interesting stuff here. All right, so the next thing is, here's a slide just to give you an idea of what we're talking about with the cultures. Or I'll get myself out of the way. So basically, when you have a vertical culture medium, up at the top, there's going to be more oxygen. And he figured out a way that he could calibrate the gradient. So these are going to be where the obligate aerobes, bacteria that need oxygen, are going to be located. If they're obligate anaerobic, they're going to be located at the bottom of the tube. If they're facultative anaerobes, meaning that they can function anaerobically, but they prefer oxygen, they're going to be higher up. All right, so anyways, what's the relevance of all this? He found that by varying the oxygen concentrations, varying the salt in the culture medium, varying these variables, he could change the bacteria back and forth between different so-called bacterial species. A lot of these bacterial species classifications are based on how it responds to a couple different, tests, like what can it grow on, how does it stain, that sort of thing. Okay, so the ability of a bacteria to change into different species is called bacterial transmutation. And why do I find this very interesting? Because what is cancer? Cancer appears to be a bacterial eukaryotic cell functioning on oxygen, oxidative metabolism, to then switch to primarily anaerobic metabolism, metabolizing without oxygen, converting glucose through glycolysis, an anaerobic cytoplasm cycle, biochemistry cycle in the cytoplasm, rather than extending the pathway in the Krebs cycle in the mitochondrial matrix and electron transport, oxidative phosphorylation in the intermitochondrial membrane. So what I'm saying is, it appears that what happens in a cancer cell, it changes from running on oxygen to running without oxygen. And that's really interesting, because what's thought to be the mechanisms of that? Otto Warburg, he won the Nobel Prize around 1930, German biochemist, considered by some the best biochemist of the 1900s, certainly one of them. He found that when he would put cells into hypoxia, they would either die or their mitochondria would become injured, and they would transform into cancer cells and run on anaerobic metabolism. That's why a PET scan, positron emission tomography scan, is positive in a cancer patient, because these cells start taking up about 100 times more glucose. All right, so this is interesting. So why am I going through all this? Because I'm saying is, if you can go from good to bad, perhaps we can redeem a cell from bad to good. And what does this all imply? What this implies is, if you've got a cancer cell, what you want to do is try to oxygenate it as well as you can, because you can maybe get that cell possibly to start going back to oxidative metabolism, or at least you can favor the surrounding cells and the tumor microenvironment, that they'll be able to control it. As the cancer cell starts taking control and dominating over the cells around it. So this is really interesting, and it also explains why he would say you'll get more commonly right-sided heart infections than left, because the blood in the right side of the heart's not oxygenated, especially in tissue locations like the right-sided heart valves, because they're going to be hypoxic to begin with poor vascularity. On top of that, they got deoxygenated blood running around them, and that's why they get more infected. So anyways, what I'm saying is, this is part of that whole concept of you can change a cell's phenotype by changing its environment. Thus, you might be able to help heal a problem by improving the tumor microenvironment, making it more oxygenated, improving the pH, making it less acidic, more basic. And this would be also avoiding excess sodium, hypernatremia, if you will. When you eat excess dietary sodium, the sodium chloride, the chloride in excess will start to displace the bicarbonate in the blood, a pH buffer, leading to a low-grade metabolic acidosis. An acidotic tumor microenvironment, tumor milieu, favors cancer growth. An acidotic tumor milieu, tumor microenvironment, suppresses immune system function. Hypernatremic, excess sodium tumor microenvironment that can activate platelets, and the platelets will have a tendency to surround metastasizing cancer cells, which can hide those cells from the immune system. The immune system runs on braille, it feels everything. So if that metastatic cancer cell floating in the blood is coated with platelets that have been activated, they will hide it from the immune system, because the immune system tries to feel it, and all it feels is these platelets surrounding it. Then it can hide the cancer cell. Okay, so this is interesting stuff. All right, and this is some of the real clever thing he did. Figured out how to vary these gradients in a calibrated, reproducible way so he could grow out pathogenic bacteria. He also figured out, this Rosenow guy, a system of dilutions that he could use to isolate low-grade pathogens. It's really interesting stuff. Okay, so anyways, transmutation of bacteria conceptually helps one understand the metabolic theory of cancer, and how all these lifestyle interventions with a low-fat, vegan diet, no oil, low sodium, could be therapeutically beneficial potentially. It's interesting stuff. It's actually, that's an AO, academic orgasm.